The citric acid cycle, or the Krebs cycle, is a cycle that occurs in the matrix of the mitochondria. The citric acid cycle repeats itself twice for each glucose molecule because two prevate molecules are made for each glucose molecule, meaning that from one glucose molecule you get two prevate molecules.
The Krebs cycle, or the citric acid cycle, begins when an acetyl group C2 is carried by CoA after glycolysis occurs and joins with a C4 molecule. This forms citrate C6. This step produces citrate C6 per cycle.
Step 2. When citrate is formed, the substrate NAD positive is oxidized into NADH. Carbon dioxide is released into the process when citrate 6 is turned into C5. NAD positive is oxidized when NAD is again formed into C5 into C4.
C4 is replaced by a phosphate group. It transfers from ADP to ATP, 2 phosphate groups to 3 phosphate groups. This step ultimately creates ATP and C4 succinate per cycle.
Succinate C4 is then oxidized into fumarate. The 2 hydrogen atoms that came off of succinate is transferred to FAD to produce FADH2. The FADH2 is transferred to the inner membrane of the mitochondria where its electrons can be transferred into the electron transport chain. This step produces FADH2 and C4 fumarate per cycle.
C4 is then turned into oxaloacetate, along with NAD plus being turned into NADH. The oxaloacetate is then combined with the acetyl group. To continue the cycle again, this step produces NADH and oxaloacetate being put for the next cycle.
The inputs are 2 C2 acetyl groups, 6 NAD+, 2 FAD, 2 ADP plus 2 phosphorus, and the outputs are 4 CO2, 6 NADH, 2 FADH2, and 2 ATP.